Okay, so now we're going to talk about some ways to control air pollution using microorganisms. So when we're talking about the air, one of the issues with using bacteria is the fact that bacteria usually lives in aqueous environments. So we can't just directly treat air with bacteria. We first have to dissolve the contaminants in the air in water and then subject that water to bioremediation by bacteria. So in order for this to work, the contaminants have to be water soluble. There are some species of yeast, though, that produce aerial hyphae that maybe could interact directly with contaminants in the air and metabolize them without having to dissolve them in water. So that would be a good application for non-water soluble contaminants. Typically, the substances are treated are VOCs like alcohols, ketones, aldehydes, odorous substances like ammonia and hydrogen sulfide gas, and xenobiotics. Xenobiotics are organic substances that are synthesized in the chemical industry and include things like chlorinated hydrocarbons. The xenobiotics are typically less readily broken down by microorganisms as other things because they're a little less biodegradable. But typically these systems run at optimal temperatures and pH that bacteria live in. So typically temperatures between 15 to 30 degrees Celsius. Of course, in moisture in an aqueous environment with pH between 6 and 9. And of course, in order for the bacteria to be active, we need a high concentration of oxygen and a lot of nutrients available for the bacteria to grow. So we're going to talk about three different types. The first type is called a biofilter. This is just a big vessel. It's got made of, the vessel itself can be made of concrete, metal, or some really durable plastic. And it's going to have a filter medium inside, and it's going to be made up of some kind of organic material like peat or heather or bark chips. And this is going to be the substrate for the bacteria. Now the medium is going to be something that's kind of porous, and so it'll hold a lot of water. And it's also, like I said, a physical support for the bacteria to grow. It provides large surface area to volume ratio. You have a lot of high internal void spaces where it can be filled with fluid, and the bacteria can act on whatever's going through the filter. And of course, it's going to be really rich in nutrients so that it can support the growth of the bacteria. Of course, the filter has to be watered sufficiently in order to maintain the optimal internal conditions, both for the gases to dissolve, but also, for the activity of the bacteria. But if you put too much water in there, it could lead to compaction, which would reduce the efficiency. So there's a optimum of how much water you can add to the biofilter. The gas that's being treated is forced through the filter. The soluble chemicals dissolve, and then within that moisture, and then the bacteria degrades the components of the solution that you're trying to get rid of. And then what comes out, the treated gas, those components have been removed by the activity of the bacteria. So a biotrickling filter is very similar to a biofilter. It's kind of like an intermediate between biofilters and the last type of technology we're going to talk about, which is a bioscrubber. So this is, again, a vessel. It's going to hold some kind of filter medium. In this case, it's going to be some kind of inert material, not an organic material, something like a clinker, which we talked about last chapter with the concrete, or some kind of a slag material. This is going to be a much stronger material. It's not as porous. It's going to be more resistant to compaction. But you still have the microbes growing on the surface of this medium. Air is forced through the filter while water simultaneously trickles down from the top. So you have air coming in at the bottom and water trickling down at the top. And so you have this countercurrent flow established between the rising gas and the falling water. And this is going to improve the efficiency of the dilution. The biofilm community, same kind of thing in the biofilter. They're going to feed on the substances in the solution and degrade it, and then the air that is released from the system is going to have those substances removed. So if you want to monitor the process, you can directly sample the water that gets recirculated within the vessel. The process control, you can add things to the circulating liquid that would be optimal for bacterial growth and action, so nutrients, oxygen, make sure buffers to make sure the pH is at the proper pH, that kind of thing. There's an advantage to the biotrickling filter over just the regular biofilters in that they can deal with higher concentrations of contaminants, and they also have a significantly smaller footprint. However, with those advantages comes higher cost, both to purchase the actual system and to run it. So the last type we're going to talk about are bioscrubbers. This isn't truly itself a biological treatment system in that you have these stacks, kind of like your typical stacks, in which you add water to dissolve the pollutants, just like you would in a regular stack. And then that has to be, that water, so the gases that are going to be treated pass through this fine water spray and get dissolved in that water, and then that water is pools at the bottom to form a reservoir. And the contaminants, of course, are dissolved in that water in the reservoir. And then that water has to then be removed and transferred to a secondary bioreactor where the actual process of biodegradation takes place. Typically, you have some kind of active sludge system to take care of that. And we're going to talk more about sludge systems and stuff when we get into wastewater treatment. But it's just a bunch of bacteria, basically, that is going to degrade the contaminants. The process control can be achieved by monitoring the water phases, and then, of course, like we said before, adding nutrients, buffers, fresh water, oxygen, whatever you need to add to keep it going. So this is, very similar to the typical scrubbers that we've seen before with the stacks. This is just when the water gets pooled, it gets treated. Instead of getting treated chemically or something, it's getting treated in a bioreactor with microorganisms. All right, so let's talk about advantages of these biotechnological approaches over more traditional chemical approaches. So typically you have competitive capital costs, low running costs, low maintenance costs. So, it's all about the money. There's lower noise, no carbon monoxide production, which is a big deal. It avoids a high temperature requirement or explosion risk, maybe that you would have associated with incineration. And, this has got a green profile, which is always, these days, is preferential. And it's robust and tolerant of fluctuations. So that is it for this short lecture. The next lecture, we're going to talk about, instead of ways to treat waste, we're going to talk about ways to prevent the waste from being produced in the first place.